Hello everyone, welcome to Study Buddy Homeschooling. In today's session, we'll be seeing vegetative propagation in plants. So what is meant by vegetative propagation in plants? When plants grow from vegetative parts of the plant like root, stem, leaves, it is known as vegetative propagation. Here there is no seed formation. Here there is no sexual reproduction. There is no pollination, fertilization. There is no seed formation here. New plants come from the existing plant parts like roots, stem, leaves, etc. So in today's session, let's see different methods of vegetative propagation. Some of the methods that are discussed in your class 10 NCERT textbooks are stem cutting, layering, grafting, propagation through leaves, spore formation and tissue culture. So let's see each one of them in this session. First we'll move to stem cutting. As the name suggests, cutting of the stems are used for the propagation of new plants. Some of the best examples are rose and hibiscus. Stem cutting works in both herbaceous and hardwood plants, but it works best in the herbaceous plants. All you have to do is take few stems with few nodes, at least 3 or 4 nodes. What are nodes? Nodes are the place from which leaves grow. So all you have to do is take a good stem cutting with 3 or 4 nodes, and all you have to do is place them in the soil with one of the nodes covered inside the soil. So after some time, this cutting will give rise to a new plant. So this is how rose and hibiscus are generally grown in our homes, through stem cutting. Next we have layering. Layering works best in certain plants like jasmine and ixora. All you have to do is choose a nice branch which is healthy and is not infested with any insects. Choose a nice branch and make a small cut in the branch. And after making a cut, all you have to do is bend the plant and cover it up with soil and leave it for 10-15 days. What happens after 15 days is root starts growing from the cut part which is covered under the soil. Now carefully you can separate the mother plant from the newly formed part. So this part will again give rise to a new plant. Layering is relatively easy and happens naturally also sometimes. Example, jasmine. Next we'll move to grafting. Grafting is a technique where joining of two different plants is used. Here you use only two different plants but not two different species. For example, if you want to graft mango, you have two different mango plants. One whose fruits are very tasty but it is very small in size, and you have another mango plant whose fruits are very big but it's not very tasty. So in this case you can graft these two mango plants and you will get the desired feature from both the plants. So the root part which you are taking, it is known as the root stock, and the branch that you are going to graft from another mango tree, that is known as the scion. All you have to do is make cut in the root stock and the scion. Make a cut in both of these and join them together with a plaster and leave it for some time. And as this grows, you will get desired characteristic features from both the mango plants. So this grafting works well in all the fruiting trees. Most of the fruiting trees like mango, guava, chiku, lemon, grafting works well. It works well in roses also, so that you can get the desired characteristic features from both the plants. And grafting, it's a skill. Actually, not many people can do it very easily. It's a skill which only few people can do it very accurately. Next, we'll move to propagation through leaves. Bryophyllum is the best example of propagation through leaves. Here you can see a mature bryophyllum leaves. Corners of the leaf develop certain buds. These buds eventually develop adventitious roots and stem and leaf-like structure, and when they mature, these buds detach from the mother leaf and it falls down and it grows into a new plant. So this is one of the easiest method of propagation, and begonia and bryophyllum are the examples of this. Next we'll move to spore formation. Spore formation is a very common method of reproduction in the lower organisms like algae, bryophytes and pteridophytes. Bryophytes example is moss and pteridophytes is fern. So in the case of spore formation, what happens is, here you can see the picture of a frond of a fern. It's a leaf of a fern, which is also known as frond. If you turn the backside of the uh, fern, you can see certain brown dots here. These brown dots are known as sori. They contain number of small cells known as spores. So when the right conditions come out, these sori break out and release spores, and each one of these spores is capable of growing into a new plant. Here you can see another example. It's a rhizopus, which is also known as bread mold. It's a fungus. Typical to fungi, it has a hyphae which is branched, and at the end of the hyphae you have a blob-like structure known as sporangium. This sporangium contains numerous pores. So when the sporangium breaks out, these pores, which are tiny cells, they release out, they break out into the air and they fall wherever they are, and each one of these pores grows into a new fungi, new rhizopus or a bread mold. That's how they grow very quickly. Examples are moss, ferns, algae and fungi. Next we'll move to tissue culture. This is one of the common techniques followed by horticulture department people, and even in nurseries people follow tissue culture. So this tissue culture works on the principle of totipotency. What is totipotency? Ability of an individual cell to grow into a complete organism. You will have to remember that not all cells are totipotent cells. Only few of the cells can be totipotent and can give rise to the whole plant itself. So this is the technique behind tissue culture, and banana, tomato, sweet potato are best examples how they can be grown through tissue culture. So what exactly happens is, in tissue culture is, you choose certain cells from the growing parts of the plant, rapidly growing parts of the plant. Few cells from this region is extracted. Okay, you can see the isolated cells here, and they are introduced to a nutrient medium rich in all the organic and inorganic nutrients. So these isolated cells, when introduced into a nutritive medium, it begins to grow and gives rise to mass of cells. So these mass of cells is known as callus. So once it reaches the callus stage, what happens is, again you'll have to shift from here to another plate rich in plant hormones like auxins, gibberellins. All you have to do is switch the callus into another medium plate full of all the plant hormones. And once this callus is subjected to these hormones, they'll start giving rise to to root, stem and leaf formation happens. Now you can see smaller plantlets coming out, and they can be transferred into nursery beds. So in case this technique is commonly followed in nursery, where hundreds of such plantlets are easily produced in a very short period of time to be able to be supplied to the farmers very easily. If you have to grow from the seed, it is going to take a lot of time, lots of risk is being involved, being eaten by plants, insects, all this there. In the case of tissue culture, there is no risk except that you will have to maintain in a very controlled environment. You should make sure that the environment is very sterile and should be very careful in the initial stages, especially from callus till callus stage and callus till the plantlet stage, because chances of infection are very high. So it has to be maintained in a very controlled environment. But otherwise, it's very easy to produce hundreds of individual plantlets in a very short period of time, and you can get the desired characteristics also. If you choose a plant which has all the positive traits will come into all the plantlets which are coming out. So it's one of the easiest techniques, and this is how farmers mostly follow in the case of banana, tomato, so that they can get hundreds of saplings very easily. If they have to go from the seed, it is going to be taking a lot of time. So farmers prefer taking plantlets from grown through tissue culture. So if you have any doubts, you can leave your comments in the comment section. Thanks for watching, and please do subscribe to our channel. Thank you.